I'm Dr. Reena Batta. I'm working as a lecturer in Botany Department, Government PG College for Women, Sector 14, Panchkula. Today I'm going to discuss on the topic Colletotrichum. First of all, the systematic position of Colletotrichum. Colletotrichum belongs to division Eumycota, class Coelomycetes, order Blanconiales, family Blanconiaceae, and genus Colletotrichum. In this, we'll discuss about the occurrence and the habit, vegetative structure, and reproduction. As far as the occurrence and habit is concerned, this genus Colletotrichum includes several species, and most of them are facultative parasites. That means they take their nourishment from the living host, and they are mainly parasitic on higher plants, causing many diseases, commonly known as anthracnose. Now I'll tell you regarding some of the species of Colletotrichum which cause diseases in vegetables and fruits. Colletotrichum falcatum cause red rot of sugarcane. Colletotrichum atramentarium causes black dot rot of tomato. Colletotrichum corcorum causes anthracnose of jute. Colletotrichum graminecolum causes red leaf spot of jowar. Colletotrichum indicum causes anthracnose of cotton. Colletotrichum gliosporidis causes anthracnose of papaya and mango. Colletotrichum massai causes anthracnose of banana. Colletotrichum capsai causes ripe fruit rot and dieback of chilies, that is capsicum. Colletotrichum inamdari causes anthracnose of karunda. In this way, it causes so many diseases in different fruits and vegetables. Now we'll discuss about the symptoms of red rot of sugarcane. So this Colletotrichum falcatum cause red rot of sugarcane. Now the symptoms of this disease appear after the rainy season, and symptoms appear in the upper leaves first, and these leaves lose their color and droop slightly. The cane starts shriveling and the rind shrinks, and in this way longitudinal wrinkles appear on the cane. Midrib region of the leaves show dark reddish areas which spread lengthwise, forming blood red lesions with straw colored centers. Now you can see here in the diagram that in the midrib region these lesions appear with the straw colored centers, and in the infected stem also you can see here that longitudinal red streaks which are crossed by white patches, they appear. And if we split open the cane, you can see here the symptoms more clearly. It shows red blotches throughout the length of pith, and you can see these red blotches when you split open the cane. So these are the symptoms of red rot of sugarcane, which is caused by Colletotrichum falcatum. Now we'll study about the vegetative structure. As I've told you that this live as a parasite on the host. The mycelium of the fungus grows within the epidermal cells. So we can say the mycelium is endophytic. So it grows within the epidermal cells as it has penetrated the cuticle. Later on, it grows in the intercellular spaces as well as intracellularly in the host tissues. So it sends haustoria in the cells for getting nourishment. The mycelium consists of hyphae, which are septate. They are much branched. The hyphae can be colorless or dark colored, and they contain the oil droplets. So this is about the vegetative structure. Now we'll discuss about reproduction. The most common method of reproduction in case of Colletotrichum is asexual method. But perfect stage or the sexual stage was also observed in some forms of Colletotrichum. It was observed in Glomerella cingulate. This Glomerella cingulate, in which the perfect stage was found, belongs to Ascomycotina. Now we'll discuss about asexual reproduction, which is the most common method of reproduction in Colletotrichum. Asexual reproduction takes place by the formation of conidia. These are produced on the conidiophores in specific fruiting bodies called acervuli. These fruiting bodies appear as small black dots, dot-like structures on the surface of host. The acervuli are formed on the rind near the nodes, and these fruiting bodies, they developed just below the epidermis in the center of the lesion. So this means wherever you find lesions on the surface, the fruiting bodies are present just below the lesions, especially in the center of the lesion. Now, if we consider the fruiting body acervulus, it consists of large number of aseptate, one-celled conidiophores. You can see here in the diagram. So these conidiophores are one-celled and they are arranged in palisade-like manner. Each conidiophore develops single conidium at its apex. So in this way, large number of conidia are produced on the conidiophores, and they are present just below the epidermis. These conidia are held together by pink colored slimy mucilaginous secretion, and they are also present brown colored long 1 to 4 septate setae around the acervellus. You can see here in the diagram the setae which are 1 to 4 celled, and conidiophores are one celled, and on the conidiophores there are present conidia. Now these setae are swollen at the basis, and the tips are tapering or rounded. And as I've already told you that these conidia are held together by pink colored slimy mucilaginous secretion. And all these structures, the conidiophores, conidia and setae, these are developed just below the epidermis. And you can see here that the conidia are one-celled, short, cylindrical or slightly curved or sickle-shaped. They can be hyaline or slightly pink-colored and densely granular bodies. Now each conidium possesses a clear vacuole-like globule. So these conidia are produced in large number, approximately 30 or more, in the fruiting body acervellus. And as I've already told you, they are held together by mucilaginous substance. But when these conidiophores along with the conidia and setae exert pressure on the epidermis will rupture and these fruiting bodies are exposed, and these conidia and conidiophores are also exposed. Now these conidia get separated by rainwater and disseminated by wind-blown raindrops. So in this way, the conidia are released, and whenever they'll find a suitable substratum, each conidium germinates by giving out 1 to 4 or generally 2 germ tubes. And in this way, hyphae are developed, and this leads to formation of mycelium. So in this way, the conidium give rise to new mycelium. So this is a very common method of reproduction in case of Colletotrichum. As I told you earlier also that in few forms of Colletotrichum, especially in the glomerella cingulate, perfect stage or sexual stage was observed. And this glomerella cingulate belongs to Ascomycotina, and the perfect stage or the sexual stage in glomerella cingulate is perithecium. Perithecium is globose, or it may be flask-shaped fruiting body, and this flask-shaped fruiting body has a definite apical pore called ostiole. The fertile layer, that is called hymenium, which consists of asci and paraphysis, is situated in the basal portion of perithecium cavity. So at the basal portion of perithecium cavity, they are present asci and paraphysis. Paraphysis are sterile, whereas asci bear ascospores. Now this ostiole, which is the opening of the perithecium fruiting body, it is guarded by sterile hyphal branches called periphysis. Now these asci contain ascospores. When they will mature, the asci will burst open and these ascospores are released through the ostiole. So these ascospores are responsible for the formation of new mycelium, but this is not a very common method of reproduction in case of Colletotrichum. So the most common method of reproduction in case of Colletotrichum is asexual method. So this is all about the Colletotrichum, that how the reproduction takes place. I have already explained you. So this is all about Colletotrichum. Thank you.